Allah, the most beneficent and merciful. Here I am, Muhammad Jamal Khan, lecturer in botany, University of Education, Lahore. Dear students, Assalamu alaikum. Today we will discuss a topic that related to our elective subject, that is microbiology. So the course code of this subject is BOTN 4128 and this is lecture number 10. In this lecture we discuss about the culture media and various types of culture media that we use for the propagation of different microorganisms. So in this lecture we discuss the culture media, and then we have inoculum that we use to propagate a specific culturing microorganism. Then we have the cultivation and the growth of that microorganism. So in this we discuss some terminologies that related to our culture media. After this we have the purposes of bacterial cultivation, why we use culture media for microorganisms or for bacterial cultivation. In addition to this, we also discuss the ingredients of culture media. We use different types of ingredients to propagate different types of microorganisms, because microorganisms have different likings for their you can say for their nutrients. आखिर में we discuss about the classification of culture media. We have different types of classifications of culture media, and we classify culture media on the base of phases, on the base of their nutritional factor, and on the base of their some specialities, and those specialities refer to as the culture media as special media. So before we going to start this topic, that is culture media, we should know about some terminologies that we used in the preparation of culture media. So first of all, here we have the culture medium. Actually, culture medium is a medium or is a substance that is used to propagate microorganism, fungi, or in some sort we have algae. So simply we say that the nutrient material prepared for the growth of microorganism in the laboratory. So basically we propagate the microorganism in a manageable conditions, and we provide such type of condition only in our laboratories. So the microbial creature use medium for their propagation, and such type of medium we call it as culture medium. And culture medium prepared with different components. So after the preparation of cultural medium, if one want to grow a microorganism or a specific microorganism, so he or she should have a spore or some sort of substance that start growth in the culture medium. So that substance that we use for the propagation of microorganism, we call it as inoculum. So simply here, when we have the microbes in either form, either it is in the form of spores Either it is in the form of mycelium in the case of fungi, either it is in the form of colony in the case of bacteria. So when we introduce such source into a culture medium to initiate growth, we call it as inoculum. So if we use the mycelium, then mycelium is an inoculum. If we use the bacterial colony, then the bacterial colony is referred to as an inoculum. And if we use spore, then spore act like an inoculum. After that, culture. So basically, culture we use for the microorganism that propagate itself into a culture medium. So the microbes that grow and multiply in or on a culture medium, we refer it as culture. Cultivation is the process of propagating microorganism by providing the proper environmental conditions. These microorganisms may be a parasites, may be bacteria, or may be a virus. All generally require cultivation for detailed study. Simply, cultivation is a method of multiplying microorganisms by letting them reproduce in predetermined culture media under controlled laboratory conditions. So, here we discuss this cultivation with reference to in vivo environment and in vitro environment. When this cultivation or propagation of microorganism occur within the whole living organism, such type of cultivation we refer as in vivo environment. And if this cultivation or propagation of microorganism occur outside the living organism in a controlled condition, for example, like in slants or in petri plates, then such type of cultivation is referred as in vitro environment. So when microorganisms are cultivated in the laboratory, a growth environment called medium is used. So the microorganism growing in or on such medium form a culture, and the main purpose of the cultivation is that one want to obtain a isolated or specific microorganism. So now, why there is a need of bacterial cultivation, or what are the purposes of bacterial cultivation? So here we have some of the main purpose for cultivation or microbial cultivation. First of all, we want to grow and isolate our specific or desired microorganism. If one want to study about any bacteria or any fungi, then first he should grow that specific microorganism, and this is done after isolation, and then he will be able to study about that microorganism. For the study of any bacteria or any microbial population, one should grow it and isolate it. After that, the other purpose of cultivation is infection and contamination. Contaminants or colonizers. Basically, when we grow the microorganism in a specific culture medium, that culture medium propagate a specific microorganism. But sometimes we obtained multiple culture within a culture medium, but we have a desire of only one specific microorganism. So such type of multiple culture, we refer it as contaminants or contamination. So bacterial cultivation or microbial cultivation enables us to isolate only a specific microorganism of our need in multiple culture or in contaminants. In addition to this, bacterial cultivation help us to identify and to characterize the specific microorganism of our need. And this is all possible through microbial or bacterial cultivation. Culture media gives artificial environment stimulating the natural conditions necessary for growth of microorganisms. So there are some basic requirements for the formation of culture media. So here we have some of the components of culture media, because culture media need energy source, carbon source, nitrogen source, salts, and growth factors. On the account of all these factors, we use different components in the preparation of culture media. So here we have a water that used as a source of hydrogen and oxygen. Then we use agar, that is a solidifying agent. Then we use peptone, and basically peptone is the mixture of partially digested protein. And we use in culture media meat extract. It is also a protein degradation products. It may be a carbohydrate or inorganic salts. Basically the meat extract is a growth factor. Similarly, we use yeast extract. It is also a protein degradation product, or you can say that is also a growth factor. And in addition to this, the major component is blood or serum that enriches the media, that is used as a source for amino acids, proteins, and vitamins, particularly soluble vitamins as A, D, and E. It is also the source of carbohydrates, lipids, hormones, growth factors, and trace elements. And in normal media, it contains about 2 to 10% serum. Now we discuss one by one all these ingredients of culture media. So water is one of the major component of culture media. Basically we use water as a solvent, because different nutrients solve into this component. So basically tap water, if it have low mineral content, and we use it for the preparation of culture media. Otherwise we used a distilled or demineralized water, deionized water, for the preparation of culture media. So the next ingredient for culture media is agar. Basically we obtained it from seaweeds. for the preparation of solid media. So here we have some characteristics of agar. Basically, it's the chief component of agar is basically a long chain of polysaccharide. It also includes some of the impurities in the form of inorganic salts and some sort of proteins. It also possesses a long chain of fatty acids that inhibit the growth, and the main minerals present in agar are magnesium and calcium, that is the requirement for microbial communities or microbial population. So basically the agar is just a gelling agent in the preparation of culture media. It not means that it increases the nutrition value of the media or nutritive properties of the media. As far as concerned with the melting and solidifying points of agar, mostly agar melt at about 95 degrees centigrade and solidify only when cooled to about 42 degrees centigrade. So for the preparation of suitable gel from agar, a concentration of near about 1 to 2% is usually required, but the manufacturer instruction should be followed. Here we have the example. The gelifying property varies in different brands of agar. For example, the New Zealand agar has more gelifying capacity than Japanese agar. So agar is manufactured either in long shreds or as powders, and there may be a variation between different batches of the same brand and of agar. So the other most important ingredient is Peptone. So it is a universal ingredient for common media. So when we look at the properties or characteristics of the Peptone, basically it is a complex mixture of partially digested proteins, and these contains peptone, the protease, amino acids and inorganic salts that are mostly related to phosphates, potassium and magnesium. And it also contains certain accessory growth factors such as nicotinic acid and riboflavin. Basically, we use nitrogen as a basically we use peptone as a nitrogen source in culture media. From the standard grades of bacteriological peptone, some manufacturers supply special grades of peptone recommended for particular purposes. Here we have neopeptone, protease peptone, mycological peptone. So commercially available peptones or digest broth can be used as place of peptone. So after this we have the fourth ingredient of culture media, that is meat extract, and it is also available commercially and is known as lemco. So basically meat extract is basically used for different amino acids. The fifth one is the yeast extract, and it contains a wide range of amino acids as meat extract. So meat extract or yeast extract, we use one from these at a single time. So it also have a role in the growth factor of various microbial communities, and it also possesses inorganic salts. So yeast extract is majorly used as a comprehensive source of growth factors and may be substituted or maybe used in place of meat extract in culture media. So I already told you that we used meat extract either we use yeast extract in our culture media because both have similar kind of properties. So the next ingredient of culture media is malt extract. Basically it is a sugar. So near about 50% maltose present in this extract. It also contains starch, dextrin and glucose, and contains about 5% of proteins and protein breakdown products, and also contains a large number of mineral salts and growth factors that are very important for the preparation of culture, blood and serum. So these are used for culturing or enriching culture media. So either we use human or animal blood can be used, basically because we use 5 to 10% blood, and that is most usual concentration, and that is almost 10%. So serum is used in certain media as a nutritive or as a nutrient component. So according to our outline, here we have the classification of media. So we classify the media into three categories. One on the basis of the phases of growth media, and the second one on the basis of the nutritional factor, and the third one based on special media. So in this lecture we discuss only two. One is this basis of phases of growth media, and the second one on the nutritional factor. So in this, first of all we discuss about liquid media, then we have a glance on solid media, in the end we have a glance on semi media. After this we have a simple media of nutritional factor, then we have the complex media, and in the end we have a synthetic or chemically, semi-synthetic or chemically defined media. So let's we start from our first category of media, that is related to the phases of growth media. So here the growth media are used in either of two phases. One is liquid, that is also referred as broth. The second one is solid, that is referred as agar media, and the third one, that is we call it as semi-solid media. In addition to this, sometimes we used hybrid type of media, that is also called as biphasic media, that contains both a liquid and solid phase. So first we have a liquid media or broth media. So in this media we dissolve nutrients into water, and bacteria growth indicated by the change in color of liquid media or broth media that appear from clear to turbid. So numerous culture media have been designed, and the earliest culture media were liquid, but that media create difficulty to prepare pure culture. One thing that I added here, that the liquid media are not simply supplemented with the solidifying agent or agar. So here we have the example of liquid media, that is a nutrient broth. This nutrient broth is composed of simple peptone and a beef extract. In this broth, peptone contributes as an organic nitrogen in the form of amino acids and long fatty acids, and the beef extract provide additional vitamins, carbohydrates, salts and other organic compounds. In liquid media, profuse growth found. So it means that the number of microorganisms that is produced in liquid media are in very large number. So due to this, microorganisms may not have developed specific features or characteristics that enable us to identify and not provide discrimination between various microorganisms that is produced in liquid media. The other disadvantage of liquid media is that because due to profuse growth, mixed population may be promoted in liquid media. So it is very difficult to isolate different types of bacteria from a mixed population. In addition to this, a liquid media can be costly and difficult to work with. Here we have the uses of the liquid media. Basically, when we want to get a large number of microbial population or community, then we use the liquid media for bacterial growth, and other for preparing of bulk culture, as in the form when we require vaccine or antigen, then we use liquid media. The second one is solid media or solid agar media. So this media is usually obtained by adding a solidifying agent in the nutrient and water. So agarose is the most common solidifying agent that we used in this media. So petri dish containing the agar is referred to as agar media. So in solid media, bacteria have a very distinct morphology and colonies, and that exhibit many different characteristics or features, such as their color and their different shapes of their colonies. So we can easily identify the various colonies or various specimen of microorganism in solid agar media. So here we have the next category of culture media, that is based on nutritional factor. So on the behalf of nutritional factor, we further categorize into three forms. One is simple media, the other one is complex media, and the third one is chemically defined or synthetic media. So first of all, we discuss about the simple media. So here, so in this media, simply we use basic nutrients or components or ingredient that is required for the growth of the ordinary microorganism or organism. So in general, we used Peptone, water, nutrient broth, and nutrient agar for the preparation of media. So such type of media, because simple media are generally used as a basis to prepare enriched media. So that's the reason we also call it as basal media. So here we have the example of simple media on the base of their nutritional factor, that is Peptone water. So basically, Peptone water is used for cultivating non-fastidious microorganisms, for studying carbohydrate fermentation patterns, and for performing the indole test. So this Peptone water contains peptone as a source of carbon, nitrogen, vitamins, and minerals. And this medium can be used as a primary enrichment medium for growth of bacteria also. As you know, Peptone is a mixture of proteins and amino acids that is obtained by breaking down different natural products such as animal tissues, milk, and plants. So here in Peptone water, the function of Peptone is to provide protein source so that the microorganism can grow better. So the second form is the complex media. So in this media that contains ingredient of unknown chemical composition. So the complex media exhibit a variety of ingredients such as meat juices and digested proteins. So the exact chemical composition of these ingredients can be highly variable, although a specific amount of each ingredient is present in the media. So one common ingredient is peptone. So this is the protein taken from any of the variety of sources that has been hydrolyzed to amino acids and short peptides by treatment with enzymes and acids or alkalis. So extract, which are water soluble component of a substance, are also used in complex media. So actually the complex media is the composition of various substances whose chemical composition are unknown, or the derivation of chemical nature is unknown, but the amount has to be fixed for the preparation of specific media. So when we use unknown chemical composition compounds with specific amount in the recipe for the preparation of media, then we call it as complex media. Here we have the example of complex media, that is a Loria broth. This broth is a nutritionally rich medium primarily used for the growth of bacteria. This Luria broth is also known as algae medium or lysogeny broth and is commonly used for nutritionally rich medium for culturing bacteria. It comprises of tryptone, yeast extract and sodium chloride. So in the last we have a chemically defined or synthetic media. So this media is prepared exclusively from pure chemical substance, and their exact composition is known. So chemically defined media are used for various experimental purposes. For example, here we have, these are used for various special studies such as metabolic requirements of the experimental microorganisms. So here we have the example of synthetic defined or chemical refined media. So here we have glucose salts agar, and this glucose salt is a simple defined or chemically defined or synthetic media. Only organisms can make all their cellular components from glucose, and inorganic salts are able to grow on this media. As you know, because in this synthetic or chemically or defined media there is totally deprived from animal derived components. That also include microbial derived components such as yeast extract, and also it include the plant derived components. So this is all about for this lecture related to the culture medium and different types of culture medium. I hope you get this lecture very informative. Stay safe and healthy. Thank you. Allah Hafiz.